Hello, everyone. I'm Dr. Bruce Bloom, Chief Collaboration Officer at Helix, and I've been invited to give a presentation on the benefits and hurdles of drug repurposing at the 2024 OCNDS Family Symposium. It's my pleasure to do this. I have a long history of affiliation with drug repurposing. I'm currently managing the clinical team at Helix. We have two assets that are existing drugs that are moving into the clinic this year. I'm also the chief science officer at the Kabuki Syndrome Foundation, where we're in the midst of getting ready to apply for an initial new drug application of a repurposed drug with the FDA. Also a chief science officer at a funding organization called Goldman Philanthropic Partnerships. I sit on a number of boards, both for and nonprofit. I do some grant making, and I'm connected with a number of academic institutions. Almost all of this is around the idea of drug repurposing. So I'm going to share over a 20-year history with you some examples of drug repurposing that have worked really well, and I'll talk about how your organization at the end might get involved with drug repurposing. So let's start by defining drug repurposing. And there is no set definition around the world that everybody agrees on. So these are my definitions. I think they're adopted by most of the people that I talk to worldwide. But when I think of drug repurposing, I think about taking an approved drug or a nutraceutical that is already approved by a regulator in at least one country for a marketed patient use in at least one disease indication, and then testing it for use in a different condition, a different disease or disorder. So take a drug or a nutraceutical, and we'll define those in a second, and find a new use for them. Sometimes people talk about drug repositioning. And when I think about this, I think about taking a molecule that's drug like, but hasn't ever been approved for anything, but it's already safe in humans. And you test it in a disease for which it was not previously tested. And so these might be a lot of compounds on a pharmaceutical company's shelf where they started using it in patients and it was proven safe, but then it didn't meet its endpoints for whatever it was trying to be approved for. And it may be sitting there untested and not being used at all, or it may be that the company is trying to put it into some other disease, but you can reposition it. And I call it drug repurposing, even though it's not yet a drug, because for something to be called a drug, it has to be approved by a regulatory authority. There's other terms that people use: reprofiling, rebooting, off-label use, which we'll talk about in a little while. So when I say drug, I'm talking about a substance other than food that is regulatorily approved to prevent, diagnose, treat, or relieve symptoms of a disease or an abnormal condition through a physiological effect. And drugs can be small molecules, large molecules, they can be biologics. But when I think about drug repurposing, I'm mostly talking about small molecules, things that are typically taken by mouth, although you can repurpose drugs that are injected or given IV. But most of the drug repurposing is taking an oral drug and finding a new use for it. A nutraceutical is a biologically active substance that's not been approved by a regulatory authority for a specific disease indication or condition, but is available for human use. We sometimes call these supplements, vitamins. These molecules are just as biologically active as a lot of drugs. The difference is you can sell them without going through all the rigmarole that you have to go through to get a drug approved. Also, on the downside, you don't know that they are monitored like drugs are. When you make a drug as a drug company, you have to make sure that the drug substance is the same in every pill, the same amount, the same other additives to make a pill or a capsule. With a nutraceutical, you don't have to be that careful. And so you don't know when you're taking a nutraceutical whether you're getting exactly what's in there or that the same dose or substance from time to time. But that doesn't mean we shouldn't use them. And I'll give you some examples of where they've been used effectively. And then the shell compound is that drug like molecule that's been proven safe for human use in a clinical trial, but doesn't yet have a home in any particular condition. And therefore, it's not available for human use except in a clinical trial, or if the company that makes the molecule is willing to allow people to take it outside a clinical trial. So let's quickly just look at what the normal drug discovery pathway looks like if you were doing traditional new chemical entity drug development. So you have to start by discovering a molecule that might work, then you validate the target that it's hitting in the disease, then you optimize that lead compound to make sure that it gets to the target, that it doesn't cause a lot of side effects, that you don't have to give too big a dose. Then you start going through your preclinical development, which means you start with cell lines, hopefully that replicate what's going on with the patient in the right tissues, then through animal work, if there's an animal model available. And at the end of that, if you feel like you have something that could go into humans, you apply to a regulatory agency and you get their approval to start a clinical trial. The probability of success of those early discovery molecules is about a third. About a third of them that start out actually get to the spot where you could do a clinical trial. Then, if you do get approval to do a clinical trial, you typically start with phase one, which is helping you make sure that it's safe for human use and that you know what the right dose is to give to patients. Phase two is to see if you get an efficacy signal in a small number of patients. If that works and you've optimized your dose, you go on to a phase three clinical trial in a much larger group of patients that are not as carefully selected. And if it succeeds there, you can apply for a new drug application and get approval. That is about 13%. And so if you multiply 13% by 35%, you see that it's about 5% of molecules that start out over a dozen or 14 years get to the endpoint where they might be called a real drug. Now let's look at the advantage of doing some drug repurposing. And what you see is that it's a lot shorter, 6 to 8 years, and the probability of success is much higher to get to the ability to start a clinical trial. Now, one reason for that is these drugs have already been in humans and we know they're safe. And we know a lot about them. We know what other drugs they interact with. We know what their targets are. And therefore, we have a much shorter timeline to get from the beginning to the preclinical validation and a much higher probability of success. The other thing you want to look at here is in many cases, you can skip the phase one clinical trial or combine your phase one with phase two because it's already been in humans, and therefore you don't have to repeat the kind of toxicology and dose ranging. And in the rare disease space, sometimes you can combine your phase two and phase three clinical trials so you can shorten both the preclinical part of it and the clinical part of it. And maybe within 6 to 8 years, you can get to the chance to get a new drug application approved by a regulatory authority. So not only is it shorter, but it's much cheaper to do this. You don't have to figure out how to make the drug because that's already been done. And there's a lot of testing that doesn't have to take place because you can already have the information from the prior approvals of that drug. So, how do you identify a drug that might be useful in a particular disease? Well, sometimes you find this out by serendipity. So, Viagra and minoxidil are examples of that, where the side effect of therapy when you were doing a clinical trial turned out to be the actual reason to give it to patients. So, Viagra was a heart medicine, but it helped with erectile dysfunction. Minoxidil helped to grow hair, even though that wasn't what it was being tested for. Neonatal hemangiomas, the biology pointed to a drug called propranolol that might work by closing off little blood vessels in the skin. So a hemangioma is like a purple stain that's on the skin because there's lots of leaky little blood vessels at the end. And so use this heart medication, which was designed to close off blood vessels so that blood pressure would go up, and you can use it in these kids and just try it off label. And another thing that was an interesting discovery was the sugar molecule cyclodextrin was used as a placebo in an animal trial of Niemann-Pick type C. And when the clinical trial of these animals was done using cyclodextrin as the placebo and a molecule that was hoping to treat, a test molecule, it turned out that the cyclodextrin animals did actually better. And it turns out that cyclodextrin does cross into the brain and can help with Niemann-Pick type C. So serendipity brought that molecule to that particular rare disease. Another way of identifying drug repurposing is traditional biology. And here's a couple of examples that I'll give you. So there's a disease called autoimmune lymphoproliferative syndrome, which we abbreviate ALPS. And it's a white blood cell disease where the blood cells do not die in a normal cell death pattern, because most of our blood cells don't last very long, a couple, 3 weeks, then they die, the molecules are broken down, and the individual parts are used to make new molecules. But if you have ALPS, you have a mutation in a particular gene that causes your white blood cells, at least one kind of your white blood cells, to never die. But they stop working after a few weeks and they build up in the body, and eventually they kill the patients by crowding out the healthy spaces or causing anemia or infections. In this case, somebody discovered the gene that was causing the disease. Shortly after the gene was discovered, the pathway was elucidated, and it was a pathway that was implicated in a particular drug called rapamycin. The pathway was called mTOR, the mammalian target of rapamycin. And so it made sense to test this drug called rapamycin in this disease. So a mouse model was built of the disease. The mice were given the rapamycin. They got better. And we immediately went to a small clinical trial. And 5 out of the 6 kids in the clinical trial were completely in remission in less than 6 months. And the 6th child in mostly remission. And the kids, that was in 2006. It's now 2024, and I know some of those kids that 18 years later are taking the same dose of rapamycin and doing just fine. They're going to live normal, healthy lives. And after that, we found 5 other diseases that had mTOR as their target, and the rapamycin worked for them as well. There's another disease called familial dysautonomia, or FD, and it's a gene that causes a protein to not be made. And that protein is important for making the autonomic nervous system in a fetus and for making the autonomic nervous system work after the baby is born. Well, the kids make some protein, but just not nearly enough. And one of the labs that we worked with used nutraceuticals to upregulate the gene so that it made more of that protein. And luckily, the proteins that were made that didn't work weren't harmful. And then we found other ways to take some of those proteins that didn't work and reshape them so they would work. And over a period of about 10 years, over a dozen nutraceuticals were discovered that are used with these kids, and you can get them up to 100% of normal circulating protein. And if a mother has a baby with this condition, she can take these nutraceuticals and the baby will be born with a normal autonomic nervous system. And all of this was done with no clinical trials at all, just with physician and patient participation, because all of the molecules used in this therapy were nutraceuticals, things you could buy at the health food store. You can also do this through a process of drug screening. So you make an assay. An assay is a intracellular signal that the cell is doing something you want it to do or not doing that when you put a chemical on it, a drug or a nutraceutical. And you usually make something attached to this assay that will light up if it green if it something good is happening, or light up red if something bad is happening. And then you build all these cells that have this assay inside them, and the assay is related to whatever the condition is. So if you're trying to make more protein, the assay would show you the protein is there, for example. And then you take little miniature wells of these cells and you apply different chemicals to them, different drugs, different nutraceuticals at different doses, and you look through a microscope and you see, are there any changes? And we have this kind of high throughput screening where all this is done by robotics. And often you can get some lead compounds by looking at this. And then you validate them through low throughput confirmation. Sometimes we find out about drug repurposing through clinical observation. So a patient has more than one condition. Maybe they're asthmatic and they have a rare disease. And it turns out in some cases their asthma drug actually improves the rare disease, or possibly it could make it worse. But you learn something about the rare disease from the comorbidity and the drug that the patient takes for that. Sometimes it's just physicians struggling to find a solution. The patient and the physician, neither one of them likes to say there's nothing else we can do. And so often a physician will think hard about, well, what might help here? What would be an innocuous drug that is not likely to cause any problems? And maybe it will help either symptomatically or with the cause of the disease. Sometimes we find patients that self-treat or learn from other patients on social media, things that have worked for them or for their child or their loved one with a rare disease. And then there's a new thing just in the last 10 years or so called in silico screening. In silico is a Latin way of describing what we do with data. So doing everything with computer systems and computer platforms. And these days, there are massive amounts of data that are available that you can use. And we use those in artificial intelligence systems and machine learning systems. The great thing about these is they can do the work of thousands or millions of people over a very tiny period of time that would be almost impossible to do if humans were doing it. And that's the good news. The bad news is almost all the work that comes out of that then has to be validated by humans because the systems are not smart enough yet to do their own validation. Although with the new generative AIs that have come out, chat GPTs and the like, we're getting to the point where we may one day soon be able to reduce the amount of human interaction that's necessary when we predict which drugs might be used in which disease. I would recommend that you go take a look at something called the NCATS translator, NCATS. NCATS is a division of the NIH, and they came out with a new artificial intelligence system called Translator. And if your disease is on there, you can put the disease in and it will scan this massive knowledge base and it will give you a list of all drugs where there's some evidence in the literature that it might work for your disease. All diseases aren't on there, so I don't know if your disease is there, but it's worth taking a look. And they're adding all diseases, uh, they're adding more diseases all the time. The nice thing about this is the way that we predict that a drug might be related to a disease isn't because there's one thing in the literature that says that it should be connected to the disease. It's because there are little individual pieces of data that are in thousands of different publications that when you link them all together, they discover this new evidence chain between a disease and a drug. And often it creates new biology. And that's really an exciting thing for all of us. So, what are some of the advantages of drug repurposing? Well, it certainly can be faster to get to patients. As I already showed in the typical drug discovery chevron diagram, you can reduce the amount of time and cost. Um, Sometimes you can get to clinical trials in under 2 years. And as I mentioned before, you might be able to skip the early phase of the clinical trials where you're doing mostly dosing and safety. Physicians can use them without a clinical trial in a lot of jurisdictions. Certainly in the United States, physicians have the right to prescribe any drug for any patient at any time. So they can use something where we call off label. And the reason we call it off label is so every drug, when it's approved, has a very well defined label that says who it can be used for, how it can be used, what diseases, what doses. It's very specific. Now, a physician can take that drug and can give it to a patient for something totally different. They can give it in a different dose, they can give it in a different disease, they can give it in a different frequency. That's up to the physician to do. And often physicians do this. We know they do this in pain management, in mental health, and in pediatrics a lot because we don't have good clinical validation of drug therapies in those conditions. And often the same thing in rare diseases. And physicians and patients want to try things as long as they're safe and everybody has informed consent, then it's a useful way of finding out whether drugs will work. Um, Sometimes you can use it without any kind of clinical trial validation. Sometimes you can use it after clinical trial validation. There's also a pathway at the FDA for drug repurposing called the 505(b)(2) pathway, which is shorter, less expensive, and um, useful for companies that are trying to find a second or third use for a particular drug. Drug repurposing has some safety advantages too. We know the drugs, we know how to dose them, we know how they interact with other drugs, we know what the side effect profiles are for the most part. Sometimes we have to be careful when we use them in a new disease. Sometimes people with those diseases, for example, you would be very careful if you had a patient with a liver condition or a kidney condition where the liver and the kidney are useful in uh, managing the drug once you take it or excreting the drug from the body. So you have to be careful in those situations. But in most other situations, you're pretty sure you understand how that drug is going to interact, even if it's in a new disease. Um, some repurposing isn't in a new indication where we take an adult drug and we figure out how to give it to kids. There are some times where we need to do new toxicology studies in a rare indication with an old drug, just as I said, because we might have some uh, tissue or other reason to be really careful that this drug should be safe in the rare disease that we're trying. What other positives are there? It certainly can be cheaper. Um, Many of these drugs are already exceedingly cheap. There's a drug that gets repurposed all the time called metformin, which was approved many decades ago for type 2 diabetes, but it has lots of interactions because it changes the way the mitochondria work in cells. And a month dose of metformin costs about $3 at retail. So if this was a drug that could help you, you could see for a whole year it cost you $36 to get this drug. The downside is that repurposing these kinds of drugs like metformin have more very poor commercial viability. If I was to find that I could validate that metformin worked for an ultra rare disease, and the patients could buy it for $36 a year, but it cost me $10 million to get the approval. I'd have to sell a lot of drug for a long time at $36 a year to get my $10 million back. And if I'm a for profit pharmaceutical company, I'm unlikely to be interested in doing that. The drugs can be widely available that can be approved. And if it's not available in the United States, the FDA has an import program that can help with that. If it's successful, it's often generic and globally available. in a lot of countries. And where it's not available in a country, then there is a way to commercialize it. So if you, for example, have a drug that's approved only in Japan, and you could repurpose that drug in the United States, you'd be the only ones authorized to sell that drug in the United States. And even if it's cheap in Japan, you could increase the price in the United States to make some commercial money from the repurposing of that drug. And it's also great that there's a lot of knowledge. There's a lot of data available for research, both in the standard research and in the computerization in silico research. So let's talk a little bit about exclusivity and commercialization so that if you ever do get a drug that works, you can think about whether there's any commercial potential. So for most drugs to have really good exclusivity, you want to have what's called a composition of matter patent. In other words, you're the one that discovered how to make the drug and what the shape of it was, and therefore you have this 19-year patent where nobody else can manufacture that molecule in that shape. So even if other people could prove that it would work for other things, they can't make it yet. That's the strongest kind of intellectual property versus a method of use patent where somebody else has already figured out what the molecule looks like, but I prove that it can be used in another disease. Therefore, I'm the only one that can market it for that disease. But if I don't have the right to make the drug, it's not very helpful. And if I do have the right to make the drug because it's generic, what's to stop somebody else from buying somebody else's version of the generic drug, even if they're not marketing it for my disease? And that's called off label use again. And so I can't really charge more just because I have a method of use patent. Sometimes you can get orphan drug designation, which comes with 7 years in the United States of marketing exclusivity. That could be helpful again if there's no off label use where somebody else could buy the drug cheaper. There are cost differentials in how much it costs to make this available to patients. You could have a patient group that has an investigator led trial just leading to off label use, like we did in the autoimmune lymphoproliferative syndrome research that we did. We had a physician researcher that did that trial for us. And then over the next year, people from all over the world called him and he gave them advice on how to use this. We didn't make any money off the drug. That wasn't our goal, but people started using it. And it's now rapamycin has become the standard of care for that particular disease, even though it's not approved on the label for that disease, but everybody knows that it's useful and it's in the literature and physician to physician communication and also the patient groups in those diseases make sure everybody knows. Those can be low cost and very short time frame. As I said, we got that rapamycin for autoimmune lymphoproliferative syndrome done in less than 3 years for less than $500,000. And it probably saves about $60 million of healthcare costs a year for all the kids that are taking it. It also depends on the disease endpoints, the biomarker, and the time frame, which we'll talk about in a second. You can also have a patient group lead to an approval of a generic drug. It doesn't require a lot of money to get a drug approved, but you can do it. And then you can get full commercialization. So the last slide I want to talk about is what a patient group needs to do drug repurposing in the last minute I have with you. Here are the critical things: natural history characterized and useful. Make sure you have some validated endpoints or outcome measures and some biomarkers that would help you get to those validated outcomes that are easy and rapid to identify. Animal models of the disease that replicate the human condition is really important. Having patient cell lines and controls so you can see, does it look different in the patient line than it does in the regular cells? Nice to have is all sorts of data for in silico. So genomics, transcriptomics, metabolomics, those help with the artificial intelligence. Target and pathway identification, so we know what we're going after. The assay, if you do a high throughput screening that is sensitive and reproducible. Disease prevalence and knowledge of the heterogeneity of the disease. and finally, a physician researcher that can do the clinical care. I hope to answer your questions. It's been a pleasure to talk to you.